This is GATE 2022 Life Sciences S Microbiology. Question number 70. The antimicrobial activity of vancomycin is due to the dash. A. Inhibition of nucleic acid synthesis. B. Damage to cytoplasmic membrane. C. Inhibition of cell wall synthesis. And D. Regulation of DNA supercoiling. This is the NPTEL online course reference for this question. Okay, we have solved a question related to this previously, and these are the different classes of antibiotics. So the major targets of antibiotics, as you can see, they either can inhibit, so these are the different inhibitions. So they can first inhibit cell wall inhibition, either cell wall synthesis, or they can disrupt the cell wall. Second, they can the mechanism of action is by inhibition of protein synthesis. Okay, they can inhibit protein synthesis either by targeting 30S ribosome, 50S ribosome, or they can target the tRNAs activity. Okay, tRNA activity, right? We have discussed this before. The second mechanism of the third mechanism of action is DNA topoisomerases. So DNA topoisomerases are present in both prokaryotes and eukaryotes, but in bacteria there are different types of DNA topoisomerases and gyrases, whereas in eukaryotes we have different DNA topoisomerases. So we have specifically classes of drugs, namely fluoroquinolones, which targets bacterial or prokaryotic DNA topoisomerases. Then this is very important, again we have folic acid synthesis. So in folic acid synthesis we have this folic acid is mainly seen, synthesis is mainly found in bacterial pathway, and we have this gene, we have this enzyme dihydrofolate reductase, okay, DHFR. And this folic acid synthesis, mainly the sulfonamides and DHFR inhibitors, targets these enzymes, okay, in the which are found in the synthesis of folic acid. And then we have DNA damage. Fifth class is DNA damage. Then these are the imidazoles mainly, which targets bacterial DNAs. And then we have mRNA synthesis inhibitors. These classes of inhibitors, they restrict the mRNA synthesis mainly in bacteria. Okay, in cell wall synthesis we have penicillin, which are the beta lactamase inhibitor. Okay, so the beta lactam is a ring. Okay, this is a beta lactam ring, and this is mainly found in gram positive bacteria. Okay, cell wall of gram positive bacteria. So these are the classes of different penicillin like drugs. So currently all the bacteria are quite resistant to penicillins. Okay, then we have generated different other variation of penicillin, okay, which are mainly reactive to the bacteria. Okay, and then we have cephalosporins. Cephalosporins also target beta lactam. Okay, these also target beta lactam, and we have also generated first generation, second, third, fourth. So we do these are the different generations, and these are developed because of antibiotic resistance. And then we have in cell wall synthesis no lactam. So we have different glycopeptides. Okay, these glycopeptides include vancomycin and other glycopeptides. So what happens, I'll explain you in detail about vancomycin. So the mechanism of action is very simple. Okay, so what happens, we have this vancomycin antibiotic. Vancomycin, as you can see, it binds to the D-alanine and D-alanine. Okay, these are the D-alanine residue, D-ala, and this is also D-alanine. Okay, these binds to the D-alanine residue and they prevent the transglycosylation and transpeptidization. So this is the bacterial cell wall, and it has the gram positive. So they have glycosylation and different. So we have a glycosylation and peptidyl transferase activity here, where the glycosyl where the on the sugar residues, these are the sugar residues, and on these sugar residues we have at least 5 to 6 different D amino acids attached. Okay, these are the different amino acids attached on these sugar residues. So the vancomycin binds to the alanine residues. Okay, it specifically binds to the D-alanine 2 D-alanine residues, and this binding interferes transglycosylation. Okay, the transfer of glycosyl to the other transfer of the glycosyl to these sugar residues is inhibited. Okay, so and this process is known as transpeptidation. Right, transpeptidation, that is transfer of peptides. So because of vancomycin binding to these D-alanine residues, we have inhibition of transpeptidation reaction. The glycosyl units will no longer able to bind, and the whole cell wall will be destabilized, and it will lead to bacterial cell death. Okay, so vancomycin, what it does, it inhibition of cell wall synthesis.